So the Biodiversity Genomics Europe project is aiming to address biomonitoring at two different levels. The first of those is the monitoring of species, detecting what species are present and how the dynamics of those species change through time. And to do that, the project uses DNA barcoding, short, standardized reference sequences which enable the identification of individuals in a rapid and cost-effective fashion. So in Biodiversity Genomics Europe, there'll be a big focus on building the reference libraries that allow the use of these sequences in biomonitoring projects. And that can be, for instance, monitoring the change in pollinators through time, but also monitoring the change of invasive species in marine environments. But in all cases, working on the basic principles of sequencing DNA from a set of samples from the environment and comparing that to a reference library to see which species are present and how they're changing through time. And the benefit of this barcode-based identification is speed and reproducibility and comparability, working towards standardized, harmonized approaches for biomonitoring species diversity in Europe. Now, the other strand of work focuses on reference genomes. And here, the focus of interest is not tracking species through time, but enabling us to track changes in genetic diversity through time. Now, unlike species diversity, there are relatively poorly developed mechanisms for tracking changes in genetic diversity. It's complicated to do it. It isn't something that's easy to see, and there are many different ways it could be done. But the key thing of the Biodiversity Genomics Europe project is to build the reference genomes in the first place. So better characterize diversity at a genomic level, build that reference library, and then use that to underpin the development of monitoring programs that allows one to trace changes in genetic diversity through time.